Wheat is one of the most crucial cereal crops in the world, serving as a primary food source for millions of people. With the growing global population and the increasing demand for food, enhancing wheat production has become more critical than ever. However, the challenge doesn't stop at production alone. Climate change and environmental fluctuations present significant obstacles to sustainable wheat cultivation. As temperatures rise and weather patterns become less predictable, it is essential to develop new wheat varieties that are more resilient and adaptable to these changing conditions. This is where the study of genetic diversity in wheat comes into play. By analyzing the genetic variation among different wheat varieties, scientists can identify traits that help plants withstand environmental stresses such as drought or extreme temperatures. These traits can then be used to breed more robust wheat varieties, ensuring higher yields and better adaptability in diverse climates. Moreover, advances in molecular biology, such as the use of DNA markers and sequencing, offer new ways to assess and utilize genetic resources. Combining these tools with traditional agricultural practices can open new avenues for improving wheat production and securing food supplies for future generations. At Science Salah Lotfy channel, we believe that understanding the science behind these advancements is crucial for addressing the challenges ahead. In the next segment, we will dive into the details of the research that explores genetic diversity in wheat, shedding light on its potential to revolutionize agriculture. Stay tuned for more insights.
Welcome to another episode of For Science Salah Lotfy channel. Today, we are diving into a groundbreaking scientific study that investigates the genetic diversity of wheat cultivars. We would like to express our deepest gratitude to Professor Dr. Mohamed Tantawy, Professor of Plant Taxonomy and Flora at the Department of Botany, Ain Shams University, Cairo, Egypt, for sharing this scientific masterpiece with our channel. Today's episode highlights a recent study titled Genetic Diversity Analysis in Wheat Cultivars using SCoT and ISSR markers, Chloroplast DNA barcoding, and grain SEM. This study was conducted by a team of highly respected researchers, including Professor Dr. Mohamed Tantawy from Ain Shams University, Professor Dr. Mohamed Ibrahim Shehata, a molecular biology expert from the same university, Dr. Hiba Hamdi Abu Sayada, Assistant Professor of Molecular Biology, Professor Dr. Al-Safa Hassan, also from Ain Shams University, Professor Dr. Abdel Fattah Badr from Helwan University, and Dr. Fatin Y. El Mouni from Fayoum University. This important research was published in BMC Plant Biology in 2023. The manuscript was submitted on October 24, 2022, and officially accepted on March 28, 2023. Stay tuned as we explore the fascinating insights into wheat genetic diversity that could shape the future of agriculture. Thank you for watching. Make sure to like and subscribe for more scientific content.
A simple grain, yet the cornerstone of civilizations. From the ancient fields of Mesopotamia to modern bread baskets, wheat has fueled humanity's rise. Its versatility knows no bounds. Bread, pasta, noodles, and countless other staples grace tables worldwide. This humble grain is more than just sustenance. It's woven into the fabric of our cultures and economies. But a storm is brewing. Climate change, with its erratic weather patterns and rising temperatures, threatens this vital food source. Droughts parch once fertile lands while floods drown fields. Heat stress scorches delicate seedlings, diminishing yields and jeopardizing global food security. The challenge is clear. We must fortify wheat against the onslaught of climate change. The solution? Unlocking the secrets held within wheat's own genetic code. Climate change is not a distant threat, its impacts are felt here and now. Rising temperatures bring more frequent and intense heat waves, pushing wheat plants beyond their limits. Droughts grip farmlands, leaving crops thirsty and stunted. These changes in weather patterns disrupt traditional farming practices and jeopardize harvests. The consequences are far-reaching. Reduced wheat yields translate to higher food prices, impacting families and communities worldwide. Food insecurity looms large, particularly in regions heavily reliant on wheat as a staple crop. The urgency to adapt wheat to a changing climate is undeniable. To fortify wheat against climate change, scientists are delving into its genetic blueprint. Like a complex instruction manual, DNA holds the key to understanding how wheat grows, adapts, and responds to its environment. By deciphering this code, researchers can identify genes that confer resilience to drought, heat, and other climate-related stresses. Enter the world of genetic markers. These powerful tools act like signposts, highlighting regions of DNA associated with desirable traits. ISSR and SCoT markers, for instance, allow scientists to differentiate between wheat varieties based on their genetic makeup. DNA barcoding, like a genetic fingerprint, provides a rapid and accurate way to identify different wheat types. And by analyzing the surface of wheat grains, researchers can glean insights into their texture, nutritional content, and resistance to pests and diseases. Section 4. Cultivating Resilience, Breeding a Better Wheat for a Changing World. Armed with this genetic knowledge, scientists are harnessing the power of traditional breeding techniques to develop climate-resilient wheat varieties. By crossbreeding plants with desirable traits, they can combine genes that enhance drought tolerance, heat resistance, and yield. Imagine a wheat variety that thrives in arid conditions, its roots delving deep to access scarce water. Or a heat-tolerant wheat, its leaves unfazed by scorching temperatures. These are not futuristic fantasies but tangible possibilities thanks to the power of genetic research and breeding. Section 5. From Lab to Field, Translating Genetic Discoveries into Tangible Solutions. The journey from laboratory discoveries to farmers' fields is crucial for ensuring global food security. Once researchers identify promising wheat varieties, rigorous field trials are conducted in diverse environments to evaluate their performance under real-world conditions. Collaboration is key. Scientists, breeders, and farmers work together to share knowledge, test new varieties, and refine breeding strategies. By bridging the gap between research and practice, these partnerships ensure that climate-resilient wheat reaches those who need it most. Section 6. Sowing the Seeds for a Food-Secure Future. The quest to safeguard wheat, our global breadbasket, is a race against time. Climate change continues to pose unprecedented challenges, demanding innovative solutions to ensure food security for a growing population. Genetic research offers a beacon of hope. By unraveling the complexities of wheat's genetic code, we gain invaluable tools to develop resilient, high-yielding varieties. This knowledge empowers us to adapt to the changing climate, ensuring a future where wheat continues to nourish generations to come. Let us sow the seeds for a food-secure future, grounded in scientific innovation and a commitment to global collaboration. Wheat, a cornerstone of human civilization, holds a rich diversity within its seemingly uniform fields. This genetic diversity is key to developing resilient and high-yielding crops. Understanding this genetic tapestry is crucial for breeders. Molecular markers illuminate variations within DNA. These markers reveal the unique genetic identity of each wheat cultivar. This exploration holds the key to improved traits, vital for future food security. Two powerful molecular markers, ISSR and SCoT, provide valuable insights into wheat's genetic makeup. These markers target specific DNA regions, revealing variations that differentiate cultivars. Variations manifest as polymorphic bands, unique patterns when DNA fragments are separated. SCoT markers focus on regions flanking the start codon, initiating protein synthesis. Analysis reveals genetic diversity within cultivars. The genetic relationships between wheat cultivars, as revealed by ISSR and SCoT markers, can be visually represented using a dendrogram, often referred to as a Euclidean distance tree. This tree-like diagram clusters cultivars based on their genetic similarity. Cultivars with similar genetic fingerprints are grouped closer together, while those with more distinct fingerprints are placed further apart. The Euclidean distance, a measure of dissimilarity, is used to construct the dendrogram. This distance represents the overall difference in banding patterns between two cultivars. Smaller distances indicate greater genetic similarity, while larger distances signify greater genetic divergence. The resulting dendrogram provides a visual representation of the genetic relationships within the group of wheat cultivars studied. It allows breeders to quickly identify closely related cultivars, which may share desirable traits, as well as distantly related cultivars, which may offer unique genetic resources for breeding programs. Beyond fingerprinting and mapping genetic relationships, molecular markers play a crucial role in confirming the identity of wheat cultivars. This process, known as DNA barcoding, utilizes specific DNA regions or barcodes to accurately identify and distinguish between species. In the case of wheat, these barcodes provide a reliable method for cultivar identification. Specific DNA regions, known to be highly variable between species, are targeted for barcoding. By comparing the sequence of these regions in an unknown wheat sample to a database of known sequences, scientists can accurately identify the cultivar. This technology is particularly useful in seed certification programs, ensuring the purity and authenticity of seed stocks. DNA barcoding provides a rapid and accurate method for confirming the identity of wheat cultivars, ensuring the integrity of breeding programs and seed distribution networks. This technology is essential for maintaining the genetic purity of valuable wheat varieties and preventing the spread of misidentified or counterfeit seeds. While molecular markers provide powerful tools for investigating genetic diversity, traditional morphological characteristics also offer valuable insights. One such characteristic, grain surface sculpture, plays a role in distinguishing between wheat cultivars. The intricate patterns etched onto the surface of wheat grains, often invisible to the naked eye, can be revealed through microscopic examination. These patterns, formed during grain development, are influenced by a combination of genetic and environmental factors. Variations in cell arrangement, surface texture, and the presence of microscopic features, such as grooves and ridges, contribute to the unique sculpture of each wheat cultivar. Analysis of grain surface sculpture alongside molecular data provides a more comprehensive understanding of wheat diversity. These morphological observations complement genetic findings, adding another layer of information to the intricate tapestry of wheat's genetic makeup. The exploration of wheat's genetic diversity, facilitated by molecular markers and morphological observations, holds profound implications for breeding and crop improvement. By unraveling the genetic relationships between wheat cultivars and identifying those with desirable traits, breeders can make informed decisions for crossbreeding programs. Knowledge of genetic diversity allows breeders to select parent cultivars that offer a wide range of beneficial genes. This strategic selection increases the likelihood of combining desirable traits such as high yield, disease resistance, and drought tolerance in new wheat varieties. Such advancements are crucial for meeting the demands of a growing global population and ensuring food security in the face of climate change. The ongoing exploration of wheat's genetic tapestry promises to unlock new possibilities for crop improvement. By harnessing the power of molecular markers, morphological observations, and innovative breeding techniques, scientists and breeders strive to develop superior wheat varieties that will nourish future generations. Wheat is a staple food for millions around the world. It provides essential nutrients and energy. As the global population grows, the demand for wheat continues to rise. This makes it crucial to ensure a consistent and robust wheat supply. Genetic diversity plays a vital role in meeting this demand. A diverse gene pool makes wheat more adaptable to changing environments. It also helps develop varieties resistant to diseases and pests. Without genetic diversity, wheat crops become vulnerable to widespread failure. This could have devastating consequences for food security. Therefore, understanding and preserving genetic diversity is paramount for sustainable wheat production. It allows breeders to develop improved wheat varieties. These varieties can withstand various environmental challenges and meet global food demands. Scientists use genetic markers to study genetic diversity. These markers are like flags in the DNA. They help identify variations within and between different populations. Two important types of genetic markers are ISSR and SCoT. ISSR stands for Intersimple Sequence Repeat. These markers target regions in the DNA that are repetitive. Differences in the number of repeats between individuals indicate genetic variation. SCoT stands for Start Codon-Targeted Polymorphism. These markers focus on regions near genes where variations often occur. They help identify differences in the DNA sequences that control gene expression. Both ISSR and SCoT are valuable tools for assessing genetic diversity. They provide insights into the genetic makeup of different wheat cultivars. This information helps breeders select suitable parent plants. It also guides them in developing new varieties with desirable traits. In our study, we assessed the genetic diversity of wheat cultivars. We focused on varieties from northern Africa and compared them to other regions. Using ISSR and SCoT markers, we examined the DNA of these cultivars. We used 306 markers in total. This helped us understand genetic relationships between cultivars. The dual approach provided a comprehensive view of genetic diversity. Our analysis revealed interesting differences between ISSR and SCoT markers. We compared their effectiveness based on several factors. These included polymorphism information content, resolving power, and marker index. We found that SCoT markers generally exhibited higher polymorphism rates compared to ISSR markers. This means that SCoT markers were more effective at detecting genetic variations between the wheat cultivars. This higher sensitivity makes SCoT markers a potentially more powerful tool for genetic diversity studies. The polymorphism information content, resolving power, and marker index values further supported the higher efficiency of SCoT markers. These values indicate the marker's ability to differentiate between individuals and populations. The higher values observed for SCoT markers suggest greater efficiency in distinguishing between the wheat cultivars. Our study revealed significant findings about the genetic diversity of wheat cultivars. The analysis of both ISSR and SCoT data showed a lower level of genetic diversity among northern African wheat cultivars compared to those from other regions. This finding has important implications for wheat cultivation and breeding programs. The lower genetic diversity in northern African cultivars could be attributed to several factors. These include historical breeding practices, environmental conditions, and limited gene flow between populations. A narrower genetic base makes these cultivars more susceptible to diseases, pests, and climate change. This vulnerability poses a risk to food security in the region. The higher genetic diversity observed in cultivars from other regions is encouraging. However, it underscores the need to conserve and utilize this diversity effectively. Global wheat breeding programs can benefit from incorporating diverse germplasm. This will contribute to developing more resilient and high-yielding wheat varieties. Our findings have significant implications for future wheat breeding programs. The lower genetic diversity in northern African wheat cultivars highlights the urgent need for strategies to enhance genetic diversity. Introducing new genetic material from diverse sources is one approach. This can be achieved through germplasm exchange programs and gene banks. Incorporating new genes can increase the genetic diversity of northern African wheat. Conservation efforts are also crucial. Preserving traditional varieties and land races is essential. Molecular markers like ISSR and SCoT are vital for efficient breeding. They help identify desirable genes and track their inheritance. Embracing these strategies enhances genetic diversity worldwide. This will develop resilient wheat varieties and ensure global food security. Welcome to today's video. We'll be diving into a key aspect of wheat genetic research, analyzing the diversity among 12 wheat cultivars using ISSR and SCoT markers. Let's take a closer look at the data and what it reveals about wheat genetics. Starting with the ISSR markers, researchers used 12 primers to examine genetic diversity in the wheat cultivars. These markers are crucial for understanding the genetic makeup and potential of different wheat strains. These primers were synthesized by HVD Egypt, a leading company in genetic research and biotechnology. Here's what they found. Primer ISSR 13 had the highest number of total amplicons, 26, and recorded a polymorphism percentage of 65%. This high level of polymorphism indicates a significant genetic variation among the wheat cultivars. Its ability to differentiate cultivars made it a valuable marker. This differentiation is essential for breeding programs aiming to improve wheat varieties. On the other hand, ISSR 12 had a low percentage of polymorphism at just 13%, indicating limited variation in this specific DNA region. This suggests that ISSR 12 may not be as useful for identifying genetic differences. Overall, the average polymorphism across the ISSR markers was 46.5%, providing a balanced representation of genetic diversity. This average is crucial for understanding the overall genetic landscape of the wheat cultivars studied. Next up, we have the SCoT markers, which stand for Start Codon Targeted Markers. These markers generated an even higher average polymorphism of 50%, reflecting their effectiveness in detecting genetic variation across different samples. This high level of polymorphism is crucial for identifying genetic diversity. primer SCoT 5 was particularly powerful, generating 27 amplicons and revealing 81% polymorphism, making it the standout in distinguishing between the cultivars. This high polymorphism rate indicates a strong ability to differentiate genetic variations. In contrast, SCoT 28 produced fewer amplicons and showed only 11% polymorphism, highlighting its limited capacity in this specific analysis. This lower polymorphism suggests it is less effective in detecting genetic differences. SCoT markers overall showed slightly better performance than ISSR markers, particularly in terms of resolving power and marker index. This makes them a valuable tool in genetic research, especially for studies requiring high precision and accuracy. By combining the data from both ISSR and SCoT markers, researchers were able to delve deeper into the genetic makeup of these wheat cultivars. This comprehensive view of genetic diversity is essential for understanding how different wheat varieties respond to various environmental conditions. Such insights are crucial for developing improved wheat varieties that are not only high-yielding, but also resilient to environmental stresses and diseases. The work underscores the immense value of molecular markers in modern breeding programs, enabling more precise and efficient selection processes. These ongoing efforts are vital for enhancing wheat production worldwide, ensuring food security for a growing global population. Thanks for watching. If you enjoyed this video, make sure to like, subscribe, and share it with your friends and colleagues. See you next time as we continue to explore more fascinating and groundbreaking research in the field of plant science.
Welcome to For Science Salah Lotfy channel. At the end of today's episode, For Science Salah Lotfy channel would like to thank all the researchers involved in the research for the great effort that was made. This remarkable research is the result of collaboration between top scientists from Egypt. The study, titled Genetic Diversity Analysis in Wheat Cultivars Using SCoT and ISSR Markers, Chloroplast DNA Barcoding and Grain SEM, was published in BMC Plant Biology in 2023. The work was initially submitted on October 24, 2022 and accepted on March 28, 2023. The researchers behind this study include Professor Dr. Mohamed Tantawy, Professor of Plant Taxonomy and Flora, Department of Botany, Ain Shams University; Professor Dr. Mohamed Ibrahim Shehata, Professor of Molecular Biology, Ain Shams University; Dr. Hiba Hamdi Abu Sayada, Assistant Professor of Molecular Biology, Ain Shams University; Professor Dr. Al-Safa Hassan, Professor of Plant Taxonomy and Flora, Ain Shams University; Professor Dr. Abdel Fattah Bada, Professor of Molecular Biology, Department of Botany and Microbiology, Helwan University; Dr. Fayton Y. El Mouni, Associate Professor of Taxonomy, Faculty of Science, Fayoum University. We extend our heartfelt thanks to these esteemed researchers for their incredible contributions to plant science. This study is not only important for wheat genetic research, but also has the potential to significantly impact global agriculture. We also want to thank you, our viewers, for tuning into For Science Salah Lotfy channel. If you enjoyed this episode and want to stay updated on more groundbreaking research, please subscribe to the channel, hit the like button, and share your thoughts in the comments below. Thank you for your support, and we look forward to seeing you in the next episode.